the supporting aspect such as regulation, facilities, financial system, bureaucracy management, information system, as well as quality of human resources, quality escalation of three Dharma perguruan tinggi is the primary target of Undip by stimulating the downstream of research outcomes that can be beneficially implemented for the society. We are also keen to improve the quantity of national and international academic publication, as well as the number of doctorates and professors in Diponegoro University.
Now, Diponegoro University has been improving in two important aspects of achievement: institutionally and student achievement. Diponegoro University has become a world-class university by being ranked 5th amongst almost 5,000 universities in Indonesia. Meanwhile, the students' achievement in both national and international scales of prestigious events are increasing. The vision to be an eminent research university is supported by the continued increase of research funding. The total research and civil service funding in 2018 is 123.7 billion rupiah. It consists of PNBP, the National Competition Research Funding, and Collaboration Research Funding. The intellectual property right becomes the indicator of significantly improved research. The patent consists of simple patent, patent, international patent, software, and invention legal letter. To support the academic process, Diponegoro University has been improving the student facilities. Thus, the students will not only get the knowledge, but also the actualization space. To the academic service, Diponegoro University has built a central computerized system that includes the service for the online study plan and online registration.
Iklim kompetisi di Undip yang sportif dan konstruktif memacu saya untuk terus berprestasi dalam akademik maupun non-akademik.
Didukung dengan fasilitas umum dan keakademikan yang lengkap,
sehingga membawa Undip masuk 10 besar universitas terbaik di Indonesia.
Undip berada di daerah beragam budaya yang harmonis.
Yang pastinya Undip lebih baik.
Undip lebih baik. Bagaimanapun, Undip pasti lebih baik.
Diponegoro University has been moving ahead to be the best university in Indonesia by providing 13 faculties of undergraduate and postgraduate. Diponegoro University has graduated 300,000 qualified alumnus. Diponegoro University also establishes vocational schools to generate a ready-to-work alumnus. Holding the fighting values of Pangaran Diponegoro, such as honest, brave, fair and attentive, Diponegoro University keeps producing excellent alumnus nationally and internationally, while also contributing in the development of knowledge, technology, art, culture and exercise.
Diponegoro University now is a rising star. We have planned to become one of university in 500 world university ranked. We have been continuously improving the supporting aspect such as regulation, facilities, financial system, bureaucracy management, information system, as well as quality of human resources, quality escalation of three Dharma perguruan tinggi is the primary target of Undip by stimulating the downstream of research outcomes that can be beneficially implemented for the society. We are also keen to improve the quantity of national and international academic publication, as well as the number of doctorates and professors in Diponegoro University.
Now, Diponegoro University has been improving in two important aspects of achievement: institutionally and student achievement. Diponegoro University has become a world-class university by being ranked 5th amongst almost 5,000 universities in Indonesia. Meanwhile, the students' achievement in both national and international scales of prestigious events are increasing. The vision to be an eminent research university is supported by the continued increase of research funding. The total research and civil service funding in 2018 is 123.7 billion rupiah. It consists of PNBP, the National Competition Research Funding, and Collaboration Research Funding. The intellectual property right becomes the indicator of significantly improved research. The patent consists of simple patent, patent, international patent, software and invention legal letter. To support the academic process, Diponegoro University has been improving the student facilities. Thus, the students will not only get the knowledge but also the actualization space. academic service, Diponegoro University has built a central computerized system that includes the service for the online study plan and online registration.
Iklim kompetisi di Undip yang sportif dan konstruktif memacu saya untuk terus berprestasi dalam akademik maupun non-akademik.
Didukung dengan fasilitas umum dan keakademikan yang lengkap.
Sehingga membawa Undip masuk 10 besar universitas terbaik di Indonesia.
Undip berada di daerah beragam budaya yang harmonis.
Yang pastinya Undip lebih baik.
Undip lebih baik. Bagaimanapun, Undip pasti lebih baik.
Diponegoro University has been moving ahead to be the best university in Indonesia by providing 13 faculties of undergraduate and postgraduate. Diponegoro University has graduated 300,000 qualified alumnus. Diponegoro University also establishes vocational schools to generate a ready-to-work alumnus. Holding the fighting values of Pangaran Diponegoro, such as honest, brave, fair and attentive, Diponegoro University keeps producing excellent alumnus nationally and internationally, while also contributing in the development of knowledge, technology, art, culture and exercise.
Diponegoro University now is a rising star. We have planned to become one of university in 500 world university rank. We have been continuously improving the supporting aspect such as regulation, facilities, financial system, bureaucracy management, information system, as well as quality of human resources, quality escalation of three Dharma perguruan tinggi is the primary target of Undip by stimulating the downstream of research outcomes that can be beneficially implemented for the society. We are also keen to improve the quantity of national and international academic publication, as well as the number of doctorates and professors in Diponegoro University.
Now, Diponegoro University has been improving in two important aspects of achievement: institutionally and student achievement. Diponegoro University has become a world-class university by being ranked 5th amongst almost 5,000 universities in Indonesia. Meanwhile, the students' achievement in both national and international scales of prestigious events are increasing. The vision to be an eminent research university is supported by the continued increase of research funding. The total research and civil service funding in 2018 is 123.7 billion rupiah. It consists of PNBP, the National Competition Research Funding, and Collaboration Research Funding. The intellectual property right becomes the indicator of significantly improved research. The patent consists of simple patent, patent, international patent, software, and invention legal letter. To support the academic process, Diponegoro University has been improving the student facilities. Thus, the students will not only get the knowledge, but also the actualization space. As for the academic service, Diponegoro University has built a central computerized system that includes the service for the online study plan and online registration.
Iklim kompetisi di Undip yang sportif dan konstruktif memacu saya untuk terus berprestasi dalam akademik maupun non-akademik.
Didukung dengan fasilitas umum dan keakademikan yang lengkap.
Sehingga membawa Undip masuk 10 besar universitas terbaik di Indonesia.
Undip berada di daerah beragam budaya yang harmonis.
Yang pastinya Undip lebih baik.
Undip lebih baik. Bagaimanapun, Undip pasti lebih baik.
Assalamualaikum warahmatullahi wabarakatuh. Good evening, ladies and gentlemen. Allow me to introduce myself. My name is Amanda, a student at the Faculty of Fisheries and Marine Science, Department of Aquatic Resources. It's precious change for me to be your host in this very special occasion. First of all, let's pray and praise Allah Subhanahu wa Ta'ala. Because of His bliss and mercy, we can join together without any obstacle and healthy condition in this world-class professor with topic Marine Invertebrates on Series 1. Let's greet and pray to our beloved Prophet Muhammad Sallallahu Alaihi Wasallam, who had brought us to the path of light. Ladies and gentlemen, to the Honorable Professor Triwinarni Agustini, MSc PhD, as the Dean of Faculty of Fisheries and Marine Science, Diponegoro University. The Honorable Dr. Suryanti, MPI, as the Head of Aquatic Resources Department. The Honorable Associate Professor Kennet Lundin, PhD, from University of Gothenburg and Natural History Museum, Sweden, as our keynote speakers. The Honorable Dr. Resi Christiana, SPI, MSI, as the Chairman of Mera Foundation, and today as our speakers. The Honorable Dr. Mizan Ardanu Asagabaldan, SPI, MSI, the Head of Study Program Marine Environmental Science from ITERA, as our speakers. The Honorable Dr. Diah Ayuningrum, SPD, MSI, from Department of Aquatic Resources, Diponegoro University, as our speakers. The Honorable Dr. Aninitya Sabdaningsih, SSI, MSI, as the moderator for today. And welcome to all of the participants. Ladies and gentlemen, before we come to the main session, let's start this event by praying first, so it will run well without any obstacle. Pray based on individual belief begins. Finish? Now, let me deliver the structure of the event for today as follows. First, opening by host. Second, hearing the national anthem of Indonesia. Third, the speech from the Dean of Faculty of Fisheries and Marine Science. Fourth, presentation and discussion about the ecology of sponges, ctenoporans, cnidarians, flatworms, and coelomorphs. Fifth, presentation and discussion about abundance, diversity, and life history of Nudibranch in Indonesia. Sixth, evaluation and closing. Ladies and gentlemen, now we will step to the next agenda. I would like to invite all of us to sit properly and hear Indonesia Raya, the national anthem of Indonesia. We're so sorry for the inconvenience. Let me talk to the committee about this. Wait for a moment, please. We're so sorry. Okay, thank you for Mr. Kuko. Okay, so for the next agenda, I would like, we like to apologize for the absence of the dean and the head of department at this moment. Because of this moment, all of the faculty members are being busy for academic activities because at that time there are some online meeting too. Thus, we continue to the next agenda. Okay, so for the next agenda, we will have photo session. Okay, so everyone, please open your camera and smile brightly. The committee will take the picture. Okay, I can see here for you who has opening the camera. Okay, please open your camera and we will have a photo session. Okay. For the committee, are all the participants open their camera, so we can move on. So we can take the picture for the documentary. Okay. So first, I would like to ask who will take the picture for our photo session.
One, two, three.
Oh, sir, I'm
Thank you.
unprepared. Okay. Okay. Thank you, sir, for taking the picture. So, okay. Okay, so we can move to the next session, sir. Okay, ladies and gentlemen, now we can move into the main point of the event, which will be led by Dr. Aninditya Sabdaningsi, SSI, MSI, as the moderator. So please, for the operator, could you please share the CV from Dr. Aninditya? So I will read the CV. Thank you, sir. Okay, so here we have a CV from Dr. Aninditya Sabdaningsi, the lecturer at Department of Aquatic Resources. And this, we can see the educational background. BSc from Department of Biology, Diponegoro University, and for MSc in Department of Aquatic Resources from Diponegoro University, and for the doctor degree is from Department of Aquatic Resources, Diponegoro University, and also the fellowship in Justus Liebig University, Gießen in Germany. And for Dr. Aninditya's expertise is marine biotechnology, and for the research interest is marine microbes, antimicrobial agent, and bioremediation agent. And here you can contact Dr. Aninditya at her email. Here we can see at the bottom. Okay, so for Dr. Aninditya, time and screen are yours. Thank you.
Okay, thank you, Manda, for your nice introduction. Assalamualaikum warahmatullahi wabarakatuh. Good evening, everyone.
Evening.
Yeah, thank you.
Good evening, Miss Anin.
The Honorable the Dean of Faculty of Fisheries and Marine Science, Professor Suginarni Agustini, and the Honorable the Head of Aquatic Resources Department, Dr. Suryanti. We would like to apologize that due to some deadlines and online meeting at this moment, and in the same time, they cannot join us here and they cannot accompany us. So the distinguished keynote speaker from University of Gothenburg and also from Gothenburg Natural History Museum, Sweden, Associate Professor Kennet Lundin, PhD. Thank you for joining with us today. The respectable all speakers in this guest lecture, Dr. Resi, Dr. Mezan, and also Dr. Dia, and all the participants, thank you for joining us today. Ladies and gentlemen, I am Aninditya Sabdanisi as your moderator for this seminar series. Actually, this guest lecture is a part of the World Class Professor Program that's sponsored by the Ministry of Education, Culture, Research, and Technology of the Republic Indonesia, collaboration with LPDP, Indonesia Endowment Fund for Education. And in this series, we will learn many kinds of marine invertebrates from expertise that come from different institutions. Therefore, this guest lecture also support of the Sustainable Goals Development, or we can say SDGs, number 4, 14, and also 17, which are quality education, life below water, and partnership for the cool. So, ladies and gentlemen, for the first speaker, we have special guest from Sweden. He is Associate Professor Kennet Lundin, PhD. He is a lecturer in Marine Science Department, University of Gothenburg, also as a senior curator in the Gothenburg Natural History Museum. He has educational background like what you can see in the slide. Yeah. He has educational background in chemistry, math, and ecology at Lund University. And then he continued to study biology at Gothenburg University for master program. And he received his doctor from Gothenburg University. His expertise is marine invertebrates, and also interest to marine invertebrates like Latyhelminthes, Nudibranchia, and so on. You can contact him by his email at kennet.lundin@pgregorian.sa, and you can also follow him at ResearchGate. So you can read his publication over there. And yeah, so this first session of guest lecture will be divided into 2 sessions. The first one is presentation. It's about 45 minutes and continued by discussion around 15 minutes. So maybe for the participants in the discussion session can type the questions in the chat box and also can directly deliver the questions by click the feature icon. So without further ado, please welcome Associate Professor Kennet Lundin. Are you there?
I'm here, yes.
Yeah,
Oh,
okay. How are you?
I'm fine. Bike, another.
Right.
Thank you, Anin.
Yeah.
I'm most honored to be able to speak to you here. Fantastic. So greetings to you all from Sweden in Scandinavia. Here we say Salam Malam, the same as a god kväll allesammans. I see my email address, kennet.lundin at should be VG region, VG region, because it's Västra Götaland, Western Gotland, this part of Sweden.
Okay, VG region.
VG region .se. So if you have any questions, don't hesitate.
Okay. Thank you. And now time is yours.
Yeah. So I'm going to talk about, as you heard, a few large groups of animals, ekor, as you call them in Indonesian, I think, of multicellular organisms, metazoans, what we call animals. The scientific sense of animals is metazoa. So I'm going to talk about the lower groups, the porifera, the comb jellies, Ctenophora, and the cnidarians, and then we get to the first crawling animals. So I'm trying to put this in a phylogenetic context. Now, why do we have these groups? How have they developed? What is the main characteristic and what has happened in this group? And then for the next session in 2 weeks, we will continue with other groups. And other speakers will have take some of them. And now we'll try to share my screen. So you won't see me for a while until we have the discussion session. So. So this is the title of my talk, Biology of Lower Metazoa, from sponges to flatworms. And I will try to speak in English that is intelligible. Even in Sweden and Scandinavia, we speak Swedish, but a lot of us speak English. So this is my museum, close to the university. The university, I'm going to talk about this a bit later, but it's a large university, one of the largest ones in Scandinavia. And the Natural History Museum here is the second largest natural history museum in Sweden. So it's really, we have 500,000 visitors. We have some in the collections, there's something between 6 and 10 million specimens. So there's a lot of things, a large collection that is used by researchers for all kinds of things, for taxonomy, for conservation, everything. So, and this is like what it sees. This my part of this. I'm living on the Swedish west coast. So this is, we have this rocky archipelago just outside, just to show you a bit. lot of these islands without any vegetation. We got lots of forest inland, but no vegetation because of the ice age. Because 10,000 years ago, this land was covered with ice. So the ice scraped away all. So they have these barren islands, just rocks. They look very smooth because the ice have been shuffling back and forth, inland ice. But in the water is very rich. There's a wide variety. There's a lot of biodiversity, and that's what I work with the biodiversity of mainly invertebrates, and to see what happens, how it changes over time, and also doing phylogeny. And some of the work I do diving, but this is not in Sweden. This is in the Philippines, actually. And if you can see what I'm looking at a nudibranch through the looking glass. Of course, this is one of my specialities is working with nudibranchs. Even if we will hear Resi, Christiana, talk about nudibranchs later on. And what you see here, this nudibranch is actually from the Swedish west coast. So we got nudibranchs that are very colorful. It's a rich, I think we have 100 species here in Sweden. Many of them are very colorful. They are aposematic, yes, like they are toxic. So they want to scare off fish. And you know, fish are very good in seeing colors. They are even better than us in seeing colors. So this is really signaling to the fish that don't eat me, I don't taste very good. So what I was telling you about, to set this in a context, the metazoa. The metazoa is us and other animals. There's organisms that have multicellular organization, that we have several cells, not only one, lots of cells with different functions, opposed to unicellular or colonial ones. So the first, this appeared mainly first 650 million years ago. Then you have what's so specific, what makes us different from the other protists. Yes, you have these special cell-to-cell junction molecules so the cells can communicate. So the organism can function as a whole. That is one specific thing. There are other things that is specific for the metazoa. Because you have this several multicellularity. This was one of the big steps in evolution, really big, because it allowed division of labor. Different cells could do different things, leading to multiple cell types. You get more complex organisms. But it's not us. There's certainly certain other groups that also have multiple cells, and those are green algae, red algae, brown algae. They also are complex and multicellular, but they never moved around. Why? Because they have photosynthesis. They are photosynthetic. They've been somewhere when they were protists. There was a single cellular algae. They fed on some cyanobacteria, and those cyanobacteria became chloroplasts, so they could photosynthesize and make sugar from water and sunlight. So they didn't have to move around. But we didn't do that. We went on a completely different path. There's unique among the organisms on this world. And this, as you can see, is a kind of the phylogenetic trees, the trees of life, part of the trees of life. We don't have the bacteria, archaebacteria. We don't have the plants. We don't have the other protists, single-celled animals like amoebas and such. We have what's in the base here, what metazoa. I don't know if you can see here. The fungi, we are not so far related. We are quite close to the fungi. It's a bit strange thought, actually. There are some similarities if you look at DNA and how we store energy in the cells. So you have in common with fungi and the mushrooms you can eat. And these also are related to choanoflagellates. So have these collar cells, but they are not multicellular or kind of loose colonies. And then they have the first metazoans, probably related to sponges, but sponges also diverged far away to something very special. They can ingest among the multicellular metazoans. They are the only ones that can ingest food in the cells by the cell walls closing and taking in food in the collar cells. They're unique with that. And then you have the ctenophorans, also very strange, archaic, early. You can see in the DNA and the sequence of the homeobox genes, there are similarities with the poriferans and the other higher metazoans, cnidarians. And then go up to the bilateria. You see in the tree to the left, the bilateria, those with two-sided symmetry. That's us. The coelomorphs was first with that. And then protostomes, flatworms, and as far as I go now. Because why we have this two sides, two just similar size with 2 eyes, 2 ears. You got that very early in the coelomorphs, but those are the first animals that started to move. The ones before us, cnidarians, they were sessile, sitting still, or they have this medusa. So there you have this biradial symmetry or radial symmetry. But as soon as you start to move, crawl around on the bottom, you must have 2 sides that are identical if you're going to move straight. Otherwise, you're going to go on the side. So there was, of course, there was selection for that, strong selection for that, very early. And bilaterians, even there are evidence for that in DNA. So that happened once. Actually, there's some ctenophores actually that are bilaterian symmetry that happened twice, or they were before us. So if we're more specific, what is a metazoan? What makes us similar to the trichoplax? You know, that thing that's looking like a bread, a hairy bread. Trichoplax is a very, very primitive animal, just less than a millimeter. You can find it in aquariums and in coral reefs. They crawl on the surface. They actually feed the whole lower side of the body functions as a gut. So as I said, the animals, metazoans, we don't photosynthesize. We can't do that if we don't have symbionts. Some of us have symbionts in the body, but basically we must eat. Ingestive heterotrophic. We have to eat to get nutrients. So most free-living metazoans ingest food through a mouth. Then you have already in sponges, have ctenophorans, cnidarians, us, they have a mouth. But some parasitic or symbiotic species like tapeworms, they can feed through the skin. They don't have a mouth anymore because they have a special skin that they can, where you have these nutrients are dissolved into the body. Now that's a special thing. That is something that evolved later. And as I said, sponges are a bit special with the pinocytosis, phagocytosis by the collar cells. These things, collars, it looks like a collar of microvilli, and when the central flagellum that creates a water current. So these microvilli kind of sieve the water for small nutrient particles. And one thing special also for metazoans is we have eggs and sperm cells. You also have eggs and sperm cells in the algae. Like in the algae, you have the sperms at night. They go swimming with 2 tails actually. So they have it. And other varieties. We have eggs and sperm. We have 2 separate sexes, or they call hermaphrodism. But you have 2 sexes because there's some stability. The idea is also in the other groups, you have two sexes. In green plants, you have two sexes because it seems like that is a kind of stable state. Then you have other things like the fungi have something completely different. When you come to embryogenesis, how we are developing as embryos, that's extremely diverse in diverse group of metazoans. Okay, so if we go back, was it 700 million years ago? There was one of those periods that the entire Earth was frozen. Snowball Earth is not those eras for a long time that because of the location of the continents, the Earth was completely frozen. It's very cold, and you have these single cell organisms. And the Earth thawed because the continents were moving in and allowing more water ocean currents. So the environment get warmer, and at that period of time you had a lot of oxygen, more oxygen than previous times, warmer times. So that made it possible for early multicellular animals, the Ediacaran fauna. You have the fossil from those. Actually, they were only feeding on bacterial films. So they would look like big carpets or some kind of look like feathers, but they were very slow. No mouth. It was like trichoplax. They were slowly feeding on bacterial films, bacterial mats. An example of that is Dickinsonia. Looks like that. But later on, something happened, and that happened evolutionary from our perspective as an explosion, rather fast. That some complex animal groups appear. Some started to eat on the other ones, and there was a quick arms race because they have this strong selection pressure. Or if someone was trying to eating with some kind of mouth or something else, just they had those who have some kind of protective structures. They survived, only those. So in a very short time, about 40 million years ago, this explosion radiation during the Cambrian, you suddenly see fossils of these fast-moving carnivorous forms with teeth. We have digging forms that go digging in the soil under the bacterial films to maybe to hide and to find food down there. So everything is moving around. You have this. They develop these scales and protective structures, and almost all of the large groups of animals that we have today, they evolved at that time. So and then the place is taken. Okay, it's there. It's hard for anything new to come. So if we move over to sponges, to one of those groups, they are sessile filter features. They're not moving around. There's actually a few carnivorous sponges in deep water, down in the deep sea. Actually, they catch animals and they eat them fast. Also, something that developed later because there's a very scarcity of food down there. 2,000 meters. Few cell types. Some freshwater sponges you can push through a sieve. So you separate all the cells, and you wait a few days, and they crawl back together again, and they can start communicate. They don't have any nerve cells. They communicate with the cells. Actually, few sponges you can actually see them move anyhow. Collar cells. That's a picture of the collar cells. There's three big classes: Calcarea, Demospongia, and the Hexatinelles. If Calcarea here, you can see this. They have this calcareous spicules. Simply, this is one zygon you can find in aquaria, in the way back in the aquaria on the back side. Simple organization, single outflow. They're just sieving the water. Calcareous algae. Also, you can find them rather shallow. And this is the big group, is the demosponges. You see the Cestospongia you have in Indonesia, these super big barrels when you go diving. 3 meters, they're bigger than me. You can find some big ones. Really old, 2,000, 2,500 years old, the oldest ones. And these Demospongia is typical for they have these calcareous spicules, and they also have the spicules are embedded in elastic spongine, the protein. And they also have lots of cytotoxic substances because they are sessile, they are sitting still, they are very, very exposed to other animals feeding on them. So that's why they must be toxic. And then some certain other animals have developed ways to cope with that toxin or even use it for themselves. I guess Resi will tell a little bit about that later on. In the Cestospongia, they usually have some kind of prawns, actually, crayfish prawns. They actually become like termites within the sponge. A queen and one male, and the rest are female workers, and they protect they live inside the sponge. And this should also tell you about, you have a lot of wonderful coral reefs in Indonesia, but you don't talk so much about the sponge reefs, because sponge reefs is usually a little bit deeper, but they're just as complex and as beautiful as the coral reefs. And also the corals are sensitive to overheating, coral bleaching, but the sponges are more resilient. So what you've seen like around Bali, they lost a lot of corals, but sponge reefs are thriving. So you get the shift towards sponges. So, and here's a kind of, I told about Cleona silata. It's a boring sponge that go into shells. It's a shell of a mussel, and actually you can see that the openings of the sponge are protruding. If you actually poke that with some needle or something, they withdraw a bit, so they can move. They can actually move because each single cell has a muscle. They have actin muscle, so they can actually move, but there's no coordinated movement. This is our typical sponges from North Atlantic. Look like this. Few scatter openings. You can also this Haliclona. You have Haliclona also in Indonesia. Looks like small trumpets or something. They are taken in water all around the body, and then small opening, cavernous opening. They have these collar cells, and then the filtered water opening in this big oxcula, the big openings. And they can also look like this. This type of sponge has symbiotic flagellates, also to helping them with get some sugar from it on the photosynthesis. Now, this is a nudibranch chewing on a sponge. We must have some nudibranchs. Algerda. And also this is also a Aegides from Tulamben, Bali, chewing on the sponges and also taking some of the toxins to get their own defense. So hexatinalids, the glass sponges, that's a very specific sponge type, because the spicules are made of silica, that is glass. It's glass, actually, the same thing as we use glass from, and also they transmit light very well. They're typically deep water living, and if you marry in Japan, it's a traditional gift that you get down this dried Venus flower basket, because they contain a pair of prawns. You see, there's a prawn up there, one male, one female. They come in as larvae, and then they get stuck in there. They can't get out. They live their entire life inside the sponge, and they help clean the sponge from inside. And they get food that comes in from the water current that is created by the sponge. But it's kind of a symbol for marriage. You stay together. You help each other. So it's a kind of a beautiful symbol to get as a wedding present. If we move on to Ctenophorans, there's also one of those earliest metazoan branches. And these are look very, very strange. Look at something that comes from another world. But this is actually one of the most important predators on the planet because they live in the open ocean, and 2/3 of the planet is actually open ocean. So they feed on copepods, smaller plankton, zooplankton, by radial symmetry. They're more not really. They have actually a bit of a biradial, so kind of in between radial symmetry and bilateral. Okay, because they often have 2 tentacles, but they can lack the tentacles as well. And they swim with a rows of stiff ciliary combs. That's why they call the comb jellies. And they don't have the stinging ctenocysts. Instead, they have these colloblasts. It's a picture of a coloblast. And they do have a nerve net, but they don't have any brain really, but they got a sensory complex in the upper aboral end. They have a mouth, they have a gut, and actually 2 anal pores in the aboral side. So just a few years ago, researchers showed that they actually do have anal pores. They are actually shitting, if I could say that. Because this is something that has developed independently within the ctenophorans. And when you go to cnidarians, there you have these typical stony corals, beautiful coral reef, Indonesian coral reef, it's a group that's lots of species, very successful. Diploblastic, there's only 2 cell layers with mesoglia as just a kind of a jelly between. They are radially, biradially. With radially symmetric, I mean, if you take it, you cut them in somewhere like a cake, you always get two half that are just the same. They got a kind of peripheral nerve net, but there's no central brain. They have a mouth and a gut, but don't have any anus. They just spit out what's left. The gas and change goes through the body wall. No gills, nothing. And they have these very special cnidocytes. They inject like in harpoon with a pressure, like in harpoon that goes through the skin, and have this special toxin that poison. And in the sea, those toxins are very often proteinaceous, protein, like a yolk, like in an egg. So if you heat it up, you denaturate, you destroy the toxin. They're very sensitive to heat in that way because the ocean is very stable. It never becomes hot in that sense. So if you want to heat up and destroy toxin, you need to heat water to 60 degrees Celsius. And this big class is Anthozoa, Cubozoa, Hydrozoa, Scyphozoa, Staurozoa. Actually, the parasitic group of singular cell Myxozoa. I won't go into those, all things. Anthozoans, those are one that makes the coral reefs. Syrian Torians, Nexacoralia, Hoctocoralia. Syrian Torians, they live on soft bottom on mud, and they come up at night. If you have to dive, you have to go on a muddy bottom at night and hover gently above it. They make this tube of mucus and clay of mud, so they can go down quickly if you disturb them. But if you're gentle, you have these wonderful long tentacles. Sea roses, we call them in Sweden. Sjörosor. And that's when you go down to the hexacorallian. That is the big group where they have corals and all that. They're called hexa, meaning 6, because they have 6 pairs of gastroderma septa. It's a kind of walls going like spokes of a wheel when you see it in a cut. If you take a cut of it, as you see on a drawing there. There's 6 pairs, and then you get new ones, but always this central basic symmetry of 6 that's called hexacorallia. And the polyps are generally smooth. And there we have the sea anemones and all the different kinds of corals. And here's the stony coral. I just want to tell you about this maybe you're not aware of that. It's a relative of you see the coral reefs. You have the shallow coral reefs. They're very colorful, where you have lots of fishes. But you also have the deep water stony corals. There's really, really huge coral formations that is on 200, 400 meters depth down on the continental shelves. And these can be, there's one up in Norway, 30 kilometers long, 30 meters high, really huge reefs actually, but it's down there along the continental shelves, along the entire Atlantic. And you have it around Sulawesi, Halmahera, this deep edge there. That's otherwise Javan Sea is too shallow. And then you have the same species actually as we have here. Desmophyllum pertusum, and you got some more also. So deep water corals have the same biodiversity of other species, and also it's just as important in creating biotopes, habitats of their own. And the Octocoralia, it's another group, big group of cnidarians, but they have should be 8 pairs. I made an error. Basically 6, but should be 8 pairs. 8 pairs of gastrodermal sera. Just remember that. I have to change that. And then you have the Alcyonians, the soft corals, and you have these big, wonderful sea fans, the Gorgonians. Those are the Octocorallians. And then you have the group Penauteloceans, sea pans. I don't know if you're aware of those. They are very common in soft bottom. They come up at night. Some nudibranchs feed on them. And then you have one big primary polyp with a stiff or bendable rock, is they call it. It's something to give them strength and stability. And then you have small feeding secondary polyps on rows on the sides to make it look like an old kind of feather pen that you have in the old days. And they have the ability to withdraw also if you get too close. But if you dive in soft bottom mud at night, you can find these big sea pen species in Indonesia. Yeah, and if we go to hydrozoans, I think I will have to speed up maybe a little bit. Hydrozoans is also a big group of cnidarians. That the long-lived polyp stages that form colonies, but they have the short-lived medusa. They could be big. They just turn up at certain times of the year. So have this generation change, and they don't have any gastroderma septa at all. Not 6, not 8. They don't have any at all. And this is a very, very big group, and they're very complicated. You can see them when you're diving as small kind of bushes, furry bushes. But you have to look really close. They look like feathers or small trees. They are also very specialized with toxins. Some toxins can be used to fighting cancer because they are cytotoxic. So it's important, and they are very interesting for medical research to find these molecules that kills, also for antibiotica and antibiotic substances to study these groups. Just a few. Here's a nudibranch specialized to feeding on a special kind of hydroid. And here's another nudibranch. You see the slender thing in the middle, especially adapted to feed in a special kind of hydroid and not to be seen by fish or something else. And these hydroids are often very stinging to touch, actually, when you're snorkeling or diving. If you see something that looks like a feather, or so, you should keep your hands off because it could be very stinging badly. And when they have this short-lived medusa form, the medusa could look like this, very different varieties. So another group of hydrozoans are the siphonophorans. These are pelagic, may look like ordinary jellyfish. The string jellyfish, these colonies gets to 30 meters. And the infamous Portuguese man of war. These are actually colonies of zooids with different ways of living. Some are feeding, some are making eggs and sperms, some are just making these floats. And we get to the Schiffozoans, the jellyfish. That is generally radially symmetric. The septa is basically four walls, gelatinous, with funnels. They got these channels in them. And then they also have these polyps, but the polyps is small, very small, and it's short-lived. And then the jellyfish, the Medusa, could live for a long time. And they have these peripheral sense organs, they're called ropalia, along the edge, along the lappets on the edge of the bell, Medusa. There we have these sensory organs that sense light, they sense movement, they sense position. And you see on the photo is lion's main jellyfish, Cyanea capillata, very common along the Swedish west coast. You have to watch out when you jump in the water. They usually don't get super big, maybe 30 centimeters wide, but up in the North Atlantic, they can get 2 meters across with tentacles 30 meters in length. So this is the biggest single solitary cnidarian species in the world. And if you happen to dive into one of those, it burns your skin for the rest of the day. Believe me, I've been doing that. But if you heat up and you can have acetic acid on, you can ease out the pain. Staurozoans, the Stork Jellyfish, have you heard of those? Very special group actually that sits on a stalk and they can crawl actually where you don't have it in the tropics, but on both sides of the tropics. You don't have in the polyp stage, and they have sticky tentacles. They're very odd. 4 or 5 centimeters maybe. This looks like something from another world. And okay, the ocean is another world, but we are depending on it. And then you have the cubozoans. Very, very toxic. Some of them, they're like cubes, 4 tentacles. Some, you know, the Irukandji syndrome. It is mostly in Australia, but turns out could be some casualties in Australia as well. It's a few species. Actually, cubozoans, they feed on fish. They feed on fast-moving prey. So the toxin adapted to that, that's nerve toxin that stuns the fish very quickly. So the fish can't swim away. So you just must kill the fish instantaneously now, and that's why they are no bigger than my nail on my little finger, and still they could kill me if I touch one. Some of these Caruca barnesi live in the mangrove forest, and when it rains, heavy rains, it gets flushed out to sea, and then you can actually, if you touch them. And Malukingi is named by a guy, Australian named King, who died from this, and they traced what it was, and they found actually this one. But not all species, some species are very harmless. So now we are getting to Bilateria. So Bilateria, the first, what we've been talking about previously has been sessile, or sitting on the bottom still, or floating in the water, or swimming in the water. But the first group of animals that started to crawl, and now we're back to the Cambrian explosion. We have Dickinsonia slowly feeding on bacteria, and then we have starting to get these forms that crawl fast, comparatively fast. And then you get bilateral body plan. You get the sensory cells in the front of your body. That's where you meet senses. That's where you meet something you're going to eat or something you're going to stay away from. Or so, but you get it. Of course, you get a selection for having sensory organs in front. They're later going to be a head with eyes, nostrils, taste. So you get the mouth on the ventral side. You get gonads in the back. And the first ones you get, the first basically most group, as far as we know now, it's a Cynacellomorphs. This very strange name, Cynacellum mixed name. because there's a small animal called Cynoturbella. Cynoturbella, you can have it on the Swedish west coast. It's a mysterious animal because this is like a living sac, very small. And you see in that vial from the exhibition at my museum, I put it there. This is the oldest specimen ever found of a Cynoturbella. So it looks a bit more just like a chewing gum, maybe. But for researchers, it's extremely interesting because if it belongs to the evolutionary line that's closest to the split, the first bilaterians, it's very interesting for many to see how organ systems developed. Maybe these substances we can use for medicine, something like that, to understand ourselves, to understand how our own bodies work. Because don't have any distinct organs. The eggs don't have any distinct places for eggs and sperm. And just they're hermaphroditic. They got a staticist. We call them the paradox worm in Swedish, paradoxmask, because we don't know because of all these struggles. But what was this? And now we know because of DNA studies. They are placed at the bottom. Another group in the Cynocerapha is the A-seals. The A-seals also earlier. They actually have a certain brain, but it's not the central brain. It's strange arches. There's a lot of wild varieties of the early brain, or just like a ring at the base of the epidermis, a thickening of nerve cells. Actually, there was the first the different varieties of early brain. They don't have a gut. Instead, they have a kind of cell mass, syncytium, where they feed the walls of the cell mass dissolve, and so the mixes with the food. And also a unique thing. They have internal fertilization. They have two sexes. They are hermaphroditic. The eggs and sperm actually, but they have internal directed fertilization. So they have a penis, a sclerotized penis. You have a uterus. You have a vagina. That's the first ones in history, probably about 560 million years ago. That's hard to find any fossil from this one, but you can trace it indirectly from DNA. So there you go. As even tapeworms have penises and vaginas, and they do a dance, an erotic dance in the intestine. Some researchers say it's a very beautiful dance. So that's one you can do when you do research. Now the last slide for me is in this split of the bilaterians, you get a big split of one group called the protostomians, and one group called the deuteriostomians. The deuteriostomians, that's led to us, chordates, vertebrates, eventually, because of the embryo stage in gastrulation looks like a small raspberry. There's a new mouth opening on the side, deuterostomia, new mouth. But in the protostomia, the opening, that remained as a mouth. But in us, that would be the anus in the mature animal. And the first ones of the protostomians is the flatworms. Free living forms actually get they have a central brain in the head. It's a compact little neuropilum. That is the first brain. They got like a nervous system. They framed shaped like a ladder, longitudinal lines with cross lines like a ladder. Gas exchange through the body wall, still no gills. They got kind of flame cells for excretion to get out the waste from the cells and get that certain pores. But usually don't have any anus. Some species have, but we're not there yet. Higher up, it's coming. And there are also some parasitic forms, cetstodes, trematodes, that is, tapeworms and that. Because like flatworms are like the perfect parasites for vertebrates. So they were sitting outside first. You can see the stages and in the mouth, and some then start to appear inside the body. And there is host changes, very complicated and magnificently adapted to us. And what we could say, there's lots of free-living small flatworms. The big ones, the polyclads, what we call flatworms, they must be very, very flat, because otherwise they can't breathe. They must have gas exchange. So if they're those who are 1 decimeter or something, they are thin leaves, because they still must have gas exchange. And they're also very toxic. And they are showing off in striking patterns and striking colors that for fish, certain fish can eat them, but most stay away. And if you have one of those flatworms in a jar with other animals, they die off because it's the toxins. Maybe they have their own toxin. In a coral reef, there's so much toxins going on everywhere. They've just been starting to find out what is happening and how we can use it for medicine and for understanding our own bodies and ourselves on the way. So now that's where I'm leaving you for now.
Okay.
So, okay.
Yeah.
Yeah.
Thank you very much, Kennet, for your great talk. There are many beautiful creatures in the ocean. Okay, then ladies and gentlemen, before we continue to the discussion session, we are lucky because now the Honorable Vice Dean of Faculty of Fisheries and Marine Science is here, join with us. Please allow me to invite Mr. Dr. Agus Trianto to give warm speech. Dr. Agus, yeah.
Okay, thank you very much, Anin. Good evening, everybody. This is my honor to welcoming all of you guys, especially Dr. Kennet Lundin from Gothenburg. Hopefully, my pronunciation not mistake.
No.
And also, okay, and thank you, and also all of speaker. Here's Dr. Bizan, Dr. Bia, and also Dr. Resi, who presented all of their expertise. First of all, our apologies we have on our dean because she has another meeting, including me. Actually, no, I have another meeting also. But however, still, I'm happy to have little time to welcoming all of you guys. First of all, thanks for your kindness to come in this webinar, especially to Dr. Kennet Lundin, and also all participants. Hopefully, this webinar is not just finished here, but this is like a gate to make a better collaboration in future. So welcome, Dr. Kennet. If you wish to join with us, collaboration about marine organisms, actually all of the keynote speaker, all of the speaker is expert in also in marine invertebrates, like Dr. Mizan on bryozoan, Dr. Anin on spawns, and Dr. Resi on cucumber, like that, and Dr. Dia, sorry, I forgot what is tunicate. Yeah, so it's mean you can work with all of them and also with the other participant here. And in the name of our dean, of course, we gladly will support the collaboration. Finally, again, thanks to the collaboration, and hopefully all of you enjoy this season, especially the presentation of Dr. Kennet. Actually, I follow just in the end, so you saw a very, very beautiful animal, even some of them quite toxic, like a small berasal. And the wonderful thing, you still keep specimen from 1949. It's very, very quite interesting, and hopefully in the future we may have a museum like in case of Dr. Kennet. Okay, thank you very much for your join in the session, and enjoy. Thank you very much.
Thank you very much, Dr. Agustianto, for your time and your opening speech here. Thank you. So, ladies and gentlemen, we can continue to the discussion session. If you guys have some question, you can type in the chat box, and also you can directly ask to the speaker with click raise hand. So in the chat box, we already had some questions from Mr. Paulo Nainggolan. He's coming from ITERA. We want to ask about hydrozoa.
If they differ in every climate.
Yes. Do hydrozoa differ in every climate?
I must say that if you look at the polyp stages, they look remarkably similar in the tropics, like they do in the temperate seas. They are very widespread, and there's a lot of species. Of course, you can see some dominating species have very large distribution. This is the same in Northern Atlantic. Similar species everywhere, where on other animals can be a lot of variation. So they are very similar, strikingly similar across the world when it comes to polyps. And then you have the Medusa stage. There's you have a difference when they appear, how they appear, when they appear, because up here in Northern Europe we have these strong seasons. So some Medusa you find in the springtime, and some in the summer, and some other species you only see in the autumn. So there's a very clear distinction. So I don't know really if there's something you can have when the monsoon change and rainy season. Oh, I just don't know, I should say. That it's more, but that's where I see the difference maybe.
Okay.
There's very few researchers that specialized in hydrozoan ecology, I should say.
Mm-hmm.
And you like you have Bugula, this one species is all over the world. We have it here, and you have it. You have it outside, the same species.
Yeah, even in Indonesia, it's tropical.
Yeah, Bugula nerutina, which you have it.
Yeah.
And that has a very interesting toxin called bryostatin. Maybe
yes,
that will become later.
Yes, Mejan will talk about it.
So it's a very important group you see globally, actually.
Okay. Next, for the next question, we have Jenny Sinaga from ITERA, also from ITERA. She talk in Bahasa, so maybe I can translate for you. She want to know about invertebrate. Maybe she mean invertebrate because she don't know. Is that only for definition, or is it taxon?
No, what you can say actually is what vertebrate. That is vertebrate is a group. There's a group of quadrupeds with vertebra. Like I have a vertebra, like akan have vertebra, and all the mammals and birds and snakes, they have vertebra. That is a group, vertebrates. But it's a practical division. The other, they don't have a vertebra. Those we call them invertebrates. But it's not. It is not. It's disparate groups. They don't have a common. So there's the rest. It was more like a practical division of animal groups.
So the next question comes from Galip.
Yeah.
Galip is student from this department. He wants to know about coral bleaching. I want to ask how big change to the sunscreen effect on surfing and diving activities to coral bleaching. And how
evidently there is some substances in sunscreen that kills corals. But then again, depends on how much is the water flow. Or are you in a kind of a pool, tide pool, and people go in there with lots of sunscreen? Maybe you get the toxin in the water, then you can have effect on the corals, I would guess. But if it's open ocean, then I think it's hardly have much effect if a few people have sunscreens on their bodies. But in any way, I think you should choose sunscreens that doesn't have these toxic substances. Definitely, I mean, you have those on the market, those non-toxic. So choose those. Maybe you pay a little bit extra, but if you care for the environment. But I should say about something about coral bleaching has been has actually happened before during what's it called, the little ice age. So and then they came back. The corals got the symbionts back. So it's something dynamic. And in the Red Sea, it's actually a very hot area, and still the corals keep the symbionts. Still they keep these symbiotic algae, dinoflagellates inside, because they are used to the heat.
Oh yeah.
And then there was some idea: could we transplant those corals in Indonesia, because or Australia, and then you are mixing species, so you can cause a big problem, because you're mixing, and there could be some invasive species coming.
Yeah, we don't know, but
I think
coral bleaching is coming from the dinoflagellates came out from the coral, right?
Yes, they get out because the coral gets stressed because high temperatures and also sediment in the water.
Yeah.
But they can quickly recover if we solve the real problem, that is human destructive activities. Try to find some ways to get over that.
Maybe next question. Anyone want to ask directly and on the camera? Okay. Hey, Mazen, you raise your hand, so you want to ask, right? Please unmute.
You unmute.
Yeah.
Okay. Hello, Mr. Kennet.
Oh, hello, Masan.
Yeah, it's been a while since we met in previous online meeting with Mero, with Resi. How are you, Mr. Kennet?
Yes, I'm fine. Oh,
okay. What time is it in Sweden?
Now it's 3:30 in the afternoon. So it's Selamat Sore day here now. So
yes.
it's coffee time here now.
Okay. Yes, yes. Okay, maybe I want to ask you maybe a little bit. I'm interested with a stenophore. With stenophore, yeah, how we recognize between stenophore maybe with maybe the cubozoa, the jellyfish. Maybe the stenophore is the same transparent body, and the cubozoa, I think, maybe it has a little tentacles and also the same transparent. Maybe how we can recognize in the field, maybe in the fields, how can we recognize with naked eye? Maybe we can distinguish. Oh, this is the stenophore, and this is the Nidaria, the cubozoa, maybe. And yes, yes. I'm sorry. And when we are in the field, are the eight ciliate, it's a comb in the stenophore,
yes.
They can see in the field or no? Because I'm really interested with stenophore. Thank you, Mr. Kennet.
Yeah. I was just thinking, I was joking, is that if you touch the cubozoa, you die. Then you know.
Yes.
If you're still alive, then you know. Okay.
They're still lucky.
Okay, that's the xenophore. Usually, actually, if you they have these comb-like, 8 comb-like furrows with these small combs that moves. They are moving.
Oh, yes.
And they are kind of stiff, so they refract the light. They're refracting light like a prism, like very, very tiny prisms. So it's a bit matter if you have a torchlight, and you just happen to have it in the right angle, then you can see this refracting light in blue and green and red. So if you have it behind or below, or you just have to try to experiment with the angle of light compared to what from your vision, and you can almost always see these furrows of the combs.
Oh, I see.
So if you have a good have a good torchlight and hold it, and you can almost always see that in the field. But if you don't have that light, just very dim light, or if it's like a general light, if it's a overcast day with clouds, and then it can be bit difficult to you don't have much that's very what's that word? You need to have good light to see the differences.
Okay, but
I know
I'm sorry, but so all stenophore is have the refracted the light. We can see that, or some stenophore they have, some stenophore they don't have.
All of them have the combs. I know that velum, velum, this is like a shaped like a band. Even those have these comb-like cilia in the middle. There's 2 ones and there's 2 on the side. So all of them have that. So I think you just to have the light to see the diffraction from the combs. But the cubozoans, sometimes the cubozoans lose their tentacles. So because I see sometimes, so you just have this little, cube, but no tentacles. Then it's could be very hard to see in the field. Also, they are very, very small.
Small, yes, the organism. Yeah. Thank you, Mr. Kennet,
yes,
for your answer.
Okay,
yes, thank you.
Okay, thank you, Dr. Mejan, for your question. And maybe we still have one question from Ruth Leodima. Ruth Leodima also come from ITERA.
Yeah, that the Immortal Jellyfish.
Yes.
Theoretically, yes, it's like that fantastic little tiny jellyfish. It's very small,
Mm-hmm.
and they can kind of rejuvenate themselves. They can seem to have this, and they've seen it on the laboratory environment, I should say, that kind of go backwards and rejuvenates, and they go back and restart the cells, and so you have cell generation starting to make new cells from go back to, they call it neoblast, the undifferentiated cell stage that some animals have, and we have embryos, fetuses. We have these neoblasts, undifferentiated starting cells, you could call them, that later turn out and then they difference in specialized cells. And that kind of jellyfish is like goes back as turn this backwards. That cells, the ones are going to be eggs or some are going to be stinging cells. They develop back into these undifferentiated cells, and then they go again, become young. So in theory, yes. I don't know if really know why they do this and how they can do it, and what is the implication for humans to know what really happens on what in genetic level.
Yeah.
What happened with the genes? Why don't they get the chromosomes we have? They get worn. That's why you have these lids on top. Every time you have this, we exchange our cells every 4 years. We are completely new cells. We're a new being. And so the chromosomes need to duplicate, and every time they do that, they get a little bit shorter. They lose the ends, the telomeres, so they get worn. That's one of the reasons we age and die. We cannot do that indefinitely. But Turritopsis has actually found some way to turn this backwards.
But do you know how long the Turritopsis for?
I don't know. I don't. You see this ability in the lab, but I haven't read any more papers on for how long time can this go on.
Okay. Yeah.
But then again, maybe all the labs that study cancer, different kind of carcinogenic, they have this culture of cancer cells from an American woman in the 50s. So her cancer cells live on forever. You just keep dividing. You just feed them with sugar and they divide. So she is now eternal if you just see her cancer cells. But she still have the rest of the DNA in her. And that's not made. I don't want to live like that on an agar plate. But we don't know. It's a wonderful thing. It's a wonderful thing. This small Cnidarians, and also a good example how much we can find from looking at these marine animals. How much they can give us. They may seem simple, but they're not.
Yes, simple, but no.
But not on the same time.
Okay, Kennet.
Yep.
Yeah, I think this is the end for the first session. So maybe the participants can understand better which species in the lower of Metazoan. Then you can continue for the second series for the higher of Metazoan. So, okay. Thank you very much, Kennet, for your time and your lecture. Yeah, maybe
you're welcome.
You can stay with us to listen the next speaker. Okay, ladies and gentlemen, we move to the second speaker. Before I invite her, please allow me to read her curriculum vitae first. Okay. Dr. Resi Christiana SP MSc is Chairman of Marine Education and Research Foundation. You can say Mero Foundation in Bali. She finished her undergraduate from Department of Aquaculture in this faculty, Faculty of Fisheries and Marine Science, Universitas Diponegoro, and she obtained her master and doctor from the Coastal Resource Management program from this department. So she is alumni from this department. And she experts in Nudibranchia and marine microbiology. She also loves to do some research in Nudibranch and marine microbiology. And you can also contact her if you want to ask something. tesichristiana.gmail@gmail.com. Okay. Please welcome Dr. Resi. Are you there?
Hello. Yes, I'm here.
Hello.
Yeah.
So like the first session, you have 40 minutes to talk, and after that we can have discussion.
Okay, thank you, Dr. Anin. So first of all, I'm very enjoy to follow the first session from Kennet. Hello, Kennet. Good evening in Sweden. Very nice to see you, and I'm very enjoy about metazoan. Yeah, actually this evening I would like to talk about Nudibranch. Nudibranch is one of part from metazoan. Yeah, so it's a small part actually. Yeah. First of all, the honorable to the Department of Aquatic Resource Management in Diponegoro University. So for all the committee that held this world class professor, yeah, for online, and also the dean, the vice dean of Fisheries and Marine Science, Dr. Agus Trianto. Good evening, my senior also. So this time is very honorable for me too, because I can see all of my colleagues and like Reoni, yeah, with Dr. Anin, also Dr. Mezan and Dr. Dia. I'm very glad to see all of you, and also all of the participants that join this webinar, yeah, this program. So let me share my screen. Okay, can you see my screen? Today I would like to talk about the abundance, diversity, and life history of Nudibranch in Indonesia. As I mentioned before, Nudibranch it is also part of metazoan. I'm sorry, can you hear my voice, Dr. Anin?
Yes, we can hear you clearly.
Yes, okay. So to continue the introduction, so I come from the Mero Foundation in the northeast coast of Bali. Kennet also already there. And my expertise or my interest is marine microbiology, biotechnology, Nudibranch, bioprospecting of marine natural product. And also current research for now, we are interested to study about taxonomy for marine biodiversity, understanding life history, ecological impact such as climate change for marine biodiversity, and also bioprospecting from marine natural product. So this is little introduction, yeah. And then here is the location of Mero Foundation in Bali. So you can go from Denpasar, it's like 2 hours, 13 minutes by car. Okay, so let's start with the session outline. We would like to study about Nudibranch, abundance and diversity, life history, food and defense, and a look into the future. So maybe Kennet is also expert in this part, so we can discuss together later in the discussion session. First of all, our blue planet. We should know about. We should understand about our blue planet. Ocean cover an area of 71% of the Earth's surface. Depth of up to 13,000 meters, where life still found. Occupy 90% of the world living space by volume. And then must still remains unexplored because of vast and difficult to access. And then based on the history, life began in the seas according to the evolutionary theory. And 80% of all life is found in the ocean. So this is, we can imagine, yeah, how diverse our organism in marine. And here is the marine habitat and diversity of life. This is, I took the picture from only Tulamben. So I can imagine maybe the habitat in the world and also in Indonesia. As you can see here, we know that the first picture is rocky area. I mean the rocky habitat. You can see under the rocky, you can find the bryozoan. Here is bryozoan, and also you can see sponge area, coral reef, and also sand slope. And in the middle is the shipwreck, one spot of shipwreck, which is the area in the Tulamben area that's very, very well known. And now there are so many marine organisms living there. And then now we go to Nudibranch. So what is Nudibranch and sea-slug fauna? Nudibranch is the group of mollusks and remarkable marine organisms that include both shelled and shelless species and have high scientific, ecological, and recreational value. As you can see in the picture, this is only an example of the specimen of Nudibranch. They are very colorful and very beautiful in the sea. That's why this animal invited the diver for like fun diving and just to have a photography. And yeah, before that, Kennet already explained to you about the very large group of metazoan. And now we go to Nudibranch. Nudibranch is a phylum of mollusks and class gastropod, and also subclass of Opistobranchia and ordo Nudibranch. And we will study about the subordo. Here is the oldest ordo is Doridina. Oldest classification is Doridina, Arminina, Dendronotina, and also Aiolidina. Nudibranch can be found in tropical or subtropical areas and in shallow water. There are about 3,000 described species of Nudibranch, and at least 40% of these have been found exclusive on the Indo-Pacific tropic. And still many species of them are unknown. And Nudibranch is a marine invertebrate predator rich in secondary metabolites, among others as antibacterial, antifungi, and also antiviral, anti-cancer, and anti-tumor. And most of them bioactive compounds that are accumulated from animals that prey, among others such as sponge, soft coral, hydroid, used as a defense from enemy attacks. Yeah, here is the map, just for imaging that where we can find Nudibranch. They are spread in the world in this area, in the tropical area. As you can see here, Indonesia is mostly in the tropical area. So we have so many kind of Nudibranch in this country. This one is one of our team research. We collect some Nudibranch from Indonesia. So here is, you can see from location 1, 2, 3, and 4 is come from Karimun Jawa, Bali, also Nusa Tenggara Timur, and also Ambon. And here is the Nudibranch. That time in my PhD, we only can find the like a big species. So that's why we found this. Maybe cannot know that very difficult to find nudibranch because the size is very small. So this one is actually this one is the kind of big nudibranch. So you can see this Phylidea and several species. I found a total like 38 species nudibranch in Indonesia. And now we continue study nudibranch in Bali, in front of Mero, actually, in front of Mero Foundation, in the total area like 40 times to 15 meter, in the depth 5 to 20 meters. We found like we can say like so many species. We in the total survey 23 times, we found 106 species, and the total individu is 635 species. As you can see here, current study in Mero Foundation 2021. We do survey every week. So you can see in the graph here, the diversity of them, and also this is the picture of some of them. So this is so beautiful. Later we will I will show you in the next photo. And then move to the next. Why nudibranch is very interesting to study than the other metazoan, because about 3,000 valid species of nudibranch are known in the world, and potentially more than 2,000 are not described yet, most from tropical water. So Indonesia is tropical water, so we are the source of nudibranch. So this is very interesting to study about them. What is nudibranch? In here, in the Nudibranch Identification book, so many SP. So I mean unknown, still only genus. Even sometime we found they don't have name. So this is our homework, yeah, to get the name of nudibranch. And here, this is the oldest classification when the scientists start working with nudibranch. They divide by, in here, they divide by four suborder like Doridacea, Arminacea, and Dendronotacea, and also Aioli Gaseoum. So the first picture that I share in my research for PhD, mostly I found Phylidea. So you can see here in the yellow part, Phylidea is only small part of nudibranch. I mean, still there are another species that maybe I don't see that. And yeah, this is more specific about them. We can see first is Aioli Gaseoum. Yeah, this is the species. Aioli Gaseoum typically have simple rhinophores without sheaths, cephalic tentacle, and serrata containing a branch of the midgut gland with a terminal cnidosac. Here is the, you can see the spot, yeah, of the serrata of Aioli Gaseoum. And the next is Dendronotidae. In here, you can see it's quite same. Dendronataceae have sheaths into which the rhinophore can contract, and the nodal edge is indicated by a row of serrata, which may be simple to complexly branched. And the next is Dorisacea. Dorisacea are generally and dorso-ventrally flattened, with the edge of the back, the notum extending out beyond the foot. So you can see here, they are like more flat, yeah, than the other, and they don't have so many serrata like the others. And next, we can see Arminacea. Arminacea lack sheath for the rhinophore and also generally lack cephalic tentacle. They are a phyrid group which have an external morphology ranging from dorid-like to form similar to Eodacea. So here is the, I put this from the reference is kind of the, how we say, evolution, yeah, kind of evolution of this animal, and then they become a nudibranch. Like Kennet told before that our the animal that we see now is like evolution from, I don't know, several thousand years ago, yeah. So you can see here is bubble shell. Sometimes people see sea hares and sapsuckers, Arminacean. This is now Arminacean, Dorid, and Dendronotid, and Aioli. So if you see this animal, they are like evolution. And also they have type of cerata, yeah. Also the cerata is different shape. And here is the anatomy of nudibranch. This animal has like a different organ and anatomy, yeah. They have like oral tentacles here, rhinophore, and also cerata, and also anus. But the other animal, they have like mantle and gills. And this is the type of cerata. As you can see, in the cerata, they have like nidosac. So many type of cerata. So they are very diverse in the sea. And this is another anatomy of nudibranch. As you can see also gills and rhinophore, and also dorsum, mantle flap. When they are walking, they can like make flapping. So it's very beautiful. And this is also another type shape of nudibranch. And then the question is how to find nudibranch in the sea. Yeah, how to find nudibranch in the sea. First of all, of course, we have to know about the dive guide. Yeah, so never dive alone, and always always notice where is your dive guide, because when you are very interested looking for, and then suddenly you don't know where you are because you are lost, where is your dive guide. And then knowledge is the key. So knowledge, it means we have to know everything about this animal, because when we go for diving, we can only stay underwater, yeah, optimum full for focus is like 1 hour maybe. So very short time, so we don't want to lose our time and also energy and also tank. So better we have to be ready before we start to looking for nudibranch. And also the size of nudibranch. We have to know the size of nudibranch. Like I told before, in my PhD, I only can find the big nudibranch because I don't really know that nudibranch is very, very small. The size is very, very small. So yeah, basically the big around 2 centimeter, but they also like 3 millimeter. So very, very small in the sea. And also gear help, of course. And don't forget to use different light of yours, lighter, yeah. So you have like red color, yellow color, so you can see the difference between nudibranch and also habitat, because this animal can has like camouflage like that. And then lack of information could be information. I mean, if you see something strange, that also could be information. Maybe that is a nudibranch. And also find the eggs. And what is the eggs? We will study next. And dive very slow, not too fast. Just slowly. And food is the key. What is the food of nudibranch? That is the key how to find nudibranch. So when you when you are diving and try to find nudibranch, don't forget to collect all this information, yeah, like size, scientific name. After diving, after collecting, directly we looking for in the book, and also data, depth, and also size, habitat details. That's very useful for scientific data. And here is the food of nudibranch. Very diverse, yeah. So they are feeding on algae, tunicate, sponge, hydroid, bryozoans, cnidarian, even soft coral, and also hard coral. Here is the picture, and the food, like tunicate, they love to eat small tunicate, and bryozoan, if same color everywhere, no attack by nudi. But if we find little spot, it's mean eaten by nudi. So, for example, if you find bryozoan, because bryozoan has a pattern, if the pattern distract or destroy, maybe there is a nudibranch there that eat the bryozoan. And also hydroid, they usually don't show themselves, but they stay downside on top of hydroids, sometimes sitting some nudi. So they are together, not only one nudi, probably 3 nudis, and also sponge, like blue sponge and red, and also algae, green algae. So the question is, one species is not feeding for all of them because they are very specific. So this is the animal who feeding on bryozoan. The name is like Ocenia, Janolus, and also Tecacera. As you can see here, this is the animal of bryozoan who feeding on bryozoan. This is like Pikachu, yeah, very beautiful. And next is feeding on hydroids. On hydroids is Phaselinid spa, no species name, and also Phaselina, Eubranchus, and also Doto grina meyeri. I saw in the previous slide from Kennet also, yeah. So this Doto sitting on hydroid, they are feeding on hydroid. And next is feeding on tunicate. For tunicate, you can have Nembrota and also Goniodoridella. This like jelly like transparent. This is tunicate, yeah. They are sitting in tunicate. And then this is nudibranch feeding on sponges like Joruna, Hypselodoris, and also Philidiella pustulosa. Most of them are like bigger than the other nudibranch. And the last is feeding on algae like Elysia marginata, Sierras elegans, and also Costaciella. Costaciella is very small, 1 millimeter probably. This size is very small. And next, understanding about the reproduction of nudibranch. This is the picture of the photos of Nembrota samberlaini. This is, you can see how they mating each other. And also this is Hypselodoris bullockii. You can see also this nudibranch mating. And also Hypselodoris trioni, this quiet common nudibranch, you can see everywhere. This is in shallow, like 5 meters. And this is when they are laying eggs. This is Dori prismatica atromarginata, spiral eggs. And also Ceratosoma tenu, this is the eggs, different color again, with the green nudibranch. So for reproduction, after mating, nudibranch laying eggs, and the diameter of eggs is this is approximately 0.24 centimeter to 3.5 62 centimeter. And the total amount of the egg is huge, as you can see, and they are in the capsule. So they have like around 1 to 9 capsule, and size of eggs is 160 micrometer. And this is the sample of the Nudibranch, Hatfield 2009, make this in the laboratory, in the culture. The name is Spurilla neapolitana, life history. So this is the start. You can see in the picture, the egg. But in the first picture is in the A is ventral view of reproductive adult faring, white arrow. You can see here the faring, and also egg mass, black arrows. This is like transparent. So this is a good example. So we can see where is the egg exactly. And then they laying egg in the B and C. Here like 5 days.
Okay, I think Resi is freezing now. Maybe we can wait her for a while. But if you want to start to ask, maybe you can type the questions in the chat box. I saw the first question come from Jenny, but maybe later on you guys can also have more questions about Nudibranch. Yeah, for several days we have trouble with the internet connection in Indonesia, Kennet. So we are really sorry if, yeah, maybe in the virtual seminar like this.
Maybe I can say a few words. I would say that Nudibranchs is a very relevant group. These snails that lost the shell, they don't have to wait to make a new shell. Instead, they found ways of being, they don't taste very good, or they can defend themselves, so they can grow very fast. And as you see, they feed on lots of animals that are sessile, living and sitting on hard bottom, hard substrate. And the Nudibranchs get specialized on a specialized, all of these are toxic with their own toxin. So that's why they have so many species of Nudibranchs, because they often get specialized on a specific kind of prey. But they help because they make new space. They like feed off because everything is covered. Every little tiny centimeter of rock is full animals trying to sit there. They are competing. But in Nudibranchs help is dynamic because it feeds on it. So they leave open space for new larvae to sit there and develop. So they help keeping up a large biodiversity. So we don't get just like a monotopic, just only rice field, just only rice and nothing else. Because if in an ecosystem where you have lots of species, it is very stable. It's resilient to against change in climate or change in from human interaction, and to get what they call ecosystem services, like fish when you go fishing. So a healthy ecosystem generally have lots of biodiversity, high diversity, and then it's strong and healthy and very productive. So Nudibranch helps this. That's why you need to know the different species, what those are the different identities, the different key players to know what they are doing. And also, if you have lots of different species, if something changes, then maybe there's some of the not so common species actually benefits.
Yeah, interesting
yeah.
for the nudibranch.
So that's why it's an important group. And also you can find these maybe compounds that is also interesting for us somehow.
Yes. Do you think that if the nudibranch eat different, I mean, like if the nudibranch eat algae and then eat sponges, is it specifically in the group? I mean, like, yeah, maybe each group only eat sponges and then the other only eat algae. Do you think is it specific?
Yeah, is it different? Sorry, I guess it's a short. Nudibranchs are predators. They eat other animals, and then there are certain groups that looks like nudibranchs. Resi call them, just for simplicity, not to make it too complicated, there are some related groups that actually feed on algae. There's the herbivores, and some of them actually are really super. They can feed on algae and use the chloroplasts. And they get virus to help them to use the chloroplasts so they can start to photosynthesize, and then they turn into what they call plantimals. They are in function like plants, but they are animals. So they are both plants and animals at the same time, because of retrovirus gets the genes from the plant into the snail.
Hello.
Hello. Okay.
I'm very sorry.
No problem.
I've been trying to keep the interest. But
Yes.
you were gone. Okay.
So, can I continue, Anin?
Yes, you can.
Until where, yeah? The last
wait.
explanation.
I think you talk the last is about the food.
Food?
No, after the food. The production.
This one, yeah?
Yes, I think.
Okay, I start with this one, yeah? Quick start. Yeah, I'm sorry, let's continue. You can hear me, Anin?
Yes, I can hear you.
Okay, so this is the sample of Nudibranch, Spurilla neapolitana. This is a good sample because we can see here like transparent from central view and also in black arrow. And then in the B, C is the picture of egg, the photo of egg. This is like I mean, this is like 5 days. 5 days, and after 70 days in the D, you can start seeing the eye spot in this white arrow, and then they start develop to become a villager, and then after 41 days, and then they are like become a juvenile after 17 days. This is like post metamorphic induction. And another sample, this is a Hexabranchus sanguinis. Each species has different time for develop. Like Hexabranchus sanguinis, dorsal view. In these photos, you can see like six days old embryo. And then in ventral view, 8 days of old embryo. And then they start veliger and then continue to juvenile. And then this is from Thorson, 1946, quite like old, because Thorson is try to describe the stage of Nudibranch. They divide by 3. One is direct development with no free veliger stage, and then lechitotrophic development with a short planktonic stage of only a few days during which no feeding is necessary, and then planktotrophic development with an obligatory feeding planktonic stage. And we go to defense. Nudibranch also have defense like gland with acid. So when the predator attack Nudibranch, Nudibranch can have acid. And also mimicry and also camouflage. They are like the same with the habitat, with the food. So the predator cannot find the Nudibranch. And also you can see here the nematocyte from the serrata. Each serrata contain nematocytes, which is this is stinging cell for predator. According to Cheng, 1964, any animal or plant that spend a portion or all of its life cycle intimately associated with a different and larger species of animal or plant, during which there is a physiological exchange, is considered a symbiont. This is explained that Nudibranch cannot live alone. So Nudibranch always together in ecosystem with where is the food where. So this Nudibranch can consume, accumulate from the food, become like polarization like pigment, and also even the compound. They use that for defense also. And the movement is normally slow on Nudibranch, less than 1 centimeter per minute. And also they can move considerably faster when disturbed. This is depend on the type of the animal. And some of them, like Hexabranchus, can swim. This is the sample of Numaila spaf as before. This is sitting in the sponge. If you see in the sea, they're like the color with this sponge. I mean, we cannot detect there is a Nudibranch sitting there. And then about identification, this is a report from the Netherlands that they describe 66 different species by DNA molecular and deep sea diving on expedition. So in here, I try to explain to you about how we describe Nudibranch. Here we can describe Nudibranch by morphological and also by molecular after collecting the sample. We can describe them on the radula by scanning electron microscope and also by DNA with the molecular approach. Here is the morphology. This is above is the old scanning electron microscope, we can they divide also nudibranch based on the radula. In the left side is sponge feeder, and the right side is anemone feeder. And this one, the last picture is nudibranch from Mero Foundation. This is the radula also from nudibranch. So we can see the morphology of radula is very different between each other. Yeah, for like anemone feeder, the radula is very smooth, and for sponge feeder, more strong. And this, based on morphological, we can see this is quite same, but they are different. They are Aioli alderi, Aioli glausa, and also Aioli papillosa. This is, if we see the morphology, they are not the same. They are different species. And also this one. Yeah. This is quite same if we don't know the species, but they are very different, even different genus in here. But here Eubranchus exiguus, Eubranchus linensis. And next is here also kind of nudibranch like Phaselina and also Fiora lineata. This is quite same, but they are different. And this is Philidia. Sometimes we see them in the sea. Probably they are the same, but the pattern is also different. So this is easy to describe by morphology because the pattern is totally different. This is Philidia and also Chromodoris. Some Chromodoris also has pattern the same with Philidia. Next, we also need to molecular identification. For example, this specimen, Aioli papillosa, also Aioli philominae. Sometimes we cannot describe by morphology, so we need DNA to identify the species different or not. And so from molecular, we are very thankful that because we can find a new species, morphology and molecular is very neat to combine so we can describe them very well. Because for now, scientists still discussion how to describe them based on genus and also until species. And this is for future, why we must know about nudibranch and how we can study about them and forward. In this, as you can see, from Dolabella auricularia, this is already well known that they can produce compound dolastatin as the cancer anti-cancer or leukemia. And then not only that, we also the microbe who living together with nudibranch, because the microbe who living together with the host can produce a great compound and potential, because nudibranch also walking everywhere and also feeding the other marine invertebrate. So this is very interesting in the future to study about this, about the potential of nudibranch. And then this is the last of my talk. Here is the theme of Mero Foundation. You can see here we can do research in the lab and also diving together with Mero team together, and thank you. Okay, I think that's all my talk about Nudibranch. Thank you for the time, and also sorry for the disturb of the internet. Yeah.
Yeah.
I give to you, Anin, the time.
Thank you, Dr. Resi,
Yeah.
for your talk, and yeah, very interesting for the Nudibranch. Yeah. And now we are moved to the discussion part. Anyone have questions? Ask Resi directly in Bahasa because she is Indonesian.
There's one in the chat. There's a question in the chat you can use on it.
Jenny asks again. She wants to know about where can I find Nudibranch, and are Nudibranch hard to find, and can we touch Nudibranch because for Cubozoa we cannot touch it. So maybe Resi, you can answer.
Yes, thank you for the question, Jenny. Yes, Jenny, we can find Nudibranch actually everywhere in various deep, like from even 2 meters until 30 or 35, we can find them everywhere. Even in the very bad condition, I mean not very nice water, you can also find Nudibranch. So it's very easy actually to find when you are diving. In Jepara, you can find, even in Semarang, you can also find Nudibranch. But the kind of species will be different between habitat and also the depth. And next question is about how we touch Nudibranch. It's okay if we touch Nudibranch, it's not the other animal. You can die or it's hard to you. It's okay to touch Nudibranch. It's no worries. Nudibranchs will have like defense for another predator like fish. When fish eat Nudibranch, Nudibranch can express and blow up the stinging sound like that. And then there is another question.
Yeah, from Chika. She is student from the Wijaya University. She want to ask you, does the difference in food consumed by Nudibranch affect the bioactive compounds contained in each individual?
Yeah. Hello, Chika. I know Chika already come to Mero. Yes, of course. Yeah, sure. Different food will be affect to bioactive compound. That's for sure, because each food has different compound and also habitat. So yes, for sure.
Okay.
And then next.
Other question from Adi Aryawan. He wants to ask, what caused the color difference in each species of Nudibranch?
Color different. I read in some reference, Nudibranch accumulated the color, the pigmentation from the food. For example, Nudibranch who's sitting on algae and also feeding on algae, the color is green. And also the other photos that I show you, Nudibranch in the sponges, there the color is exactly the same. And also in the hydroid. So they accumulate the pigment from food, but some of them they have color already like that. Maybe Kennet also can answer about the coloring of nudibranch.
Certain groups have closely related species can have similar, quite similar colors with variations, of course. Some of these lovely needs. This is a book on Swedish nudibranchs. So they are red with white tips on this. The serata, the extensions they have on the back with the stinging cnidocytes they've stolen from the hydros they're eating. They're on the white tips. But they had these so these contrasting colors, white and the red and the yellow and the black. And then some groups, you can have one species, but they are the different colors. The same species, but had different colors. So you need to do DNA, and then we don't really know what makes that wide spectrum. So there's all kinds of there's so many species of nudibranchs. So you have all these different examples. If you think up one example and it's there somewhere.
Yeah.
So some nudibranchs are cryptic. When you lift them up and you have them in a jar, they have the extreme colors and very the pattern in a very specific way. But when it sits on its food, there could be a sponge or a hydroid or something, then it disappears, maybe. Or it could be someone that it looks very, very distinct. I have these warning colorations for fish that don't meet me. It's like a warning, it's like a big sign.
Yeah.
Because this is such a big field.
Yeah. We call it like camouflage. Yeah, Kenneth, yeah. We call it camouflage. Like this, like my presentation, that we need DNA molecular to do that. They are different or not, because if only based on morphology, we cannot answer that. So until now, the scientists still describe about them and still develop how to classify nudibranch. Yeah.
Yeah, we see, and only in Sweden here you find a new species. This book is in Swedish. This was published this spring in March, and I'm the main author of it.
Yeah, this is new published, new book. Yeah.
Yes, it's a new book. Yeah, we have this series of national keys for different animal groups in Sweden, and also plants and mosses and lichens. So there's a keys in here, and those are in English.
Oh, great.
Yeah.
So I would say we know about approximately what we have, this 100 species, but you have several thousand, and there's so much to discover and so much to be described in Indonesia. It's absolutely amazing. So there's so much that need to be done.
So we need to extra work, right?
Absolutely.
To identify the nudibranch.
Yes, and I think just close to Semarang, if you go to the sea, and I think in low tide, if there's some place with stones and rocks, you just flip the rocks over. I think you can find some nudibranchs, don't you think? You see?
Yes, I found in Jepara in low tide, like 2 meters, even 1 meter, maybe I can find Joruna funebris. Joruna funebris, yeah.
But is it possible at low tide to go if you don't dive, just snorkel?
Yeah, it is possible if I'm not doing snorkeling, just walking.
Oh, well, if some of the students would like to find some nudibranchs by themselves, they still can do that if they go snorkeling, I think. Or Have you done it?
Yes, in Jepara maybe. But if you are lucky, because they are
Yes.
not always there. Yeah.
No, the same in Sweden. You need to be a bit like, it's hard just to snorkel. You almost need to have your scuba gear, but sometimes you can find.
Okay. Maybe for Jenny, if you are lucky, then you want to find the nudibranch, you just go walk to the beach and try to find in the behind of the stone like this, something like that. Okay. The next question is coming from Ruth Leodima, but I think this is the same with the previous question. She wants to ask, is it true that the nudibranch has a bright color from the food it eats? If so, what kind of food can make this happen in nudibranch, the bright color?
Yes, this is the same question. Each nudibranch has specific food. So like Alicia, here is on algae, and also like Doto, always on hydroid. So they are not mixed food. They always feeding on that food. So yeah, all of them nudibranch take the accumulate the pigmentation like that. Like Kenneth told also, sometimes we really cannot exactly, we only see algae without nudibranch like that. So very clever, yeah, nudibranch. Yeah. So all food they accumulate sponges, bryozoan, algae, hydroid, and coral.
So you mean if the nudibranch has several colors, it means that nudibranch eat some of the animals? Do you think about it?
No, I think nudibranch only eats one kind of specific food, not mixed food. But if so many color, not only from food, nudibranch also can camouflage.
Okay.
Yeah. So from them, they also has color, not only from food. Yeah.
Okay. For defense, yeah.
Yeah, for defense.
Okay. Thank you, Resi. I think I have several questions also for you for the nudibranch because I'm curious with it. Do you think that nudibranch, the water quality affects the life of the nudibranch? Because for the coral that we know, coral is very sensitive with the water quality, like the turbidity or sedimentation, something like that. Do you think that nudibranch also affected by the water quality?
This is ecological, yeah. But the things is nudibranch will be there if the food is there. Yeah. So they will never there if the food is not there. Maybe if you find nudibranch not in their food, probably the current bring them, but they will go back to the habitat where is the food there. If it's not, they will fast dying, will be die like that. So for water quality, that's also my question, because I found very specific in Jepara, which is not so good water quality, but there are so many Joruna funebris until 50 to 100. So they are same species and they are there. But we never found like Doto probably because yes, no hydroid. So in my opinion, will be based on foods like that. So if water quality, for example, hydroid cannot survive in that water quality, will not grow there. And Nudibranch also will be not there like that. So yeah, of course, water quality for food first.
Okay, got it. Do
Yeah.
you think the same, Kennet? Maybe you have another idea about it?
Yes. Please.
You should know that they have this the larval stage. You saw some of the photos that Resi showed that they have shell. They have a tiny little shell, and they can swim with these folds that has cilia. So they go with the current. It's different what you have in tropical areas. You have lots and lots of species, but usually not so many individuals. You have one individual here, one species, and you swim over. There's one specimen. There's another species. In temperate waters, you have fewer species, but lots of them. Of your one species, you can have thousands. Really mass occurrence for a short while, not for a long time. And they're dependent on the water should be like in spring when it's cold, clear water. For a short time you can have lots of nudibranchs, and then disappear. And next year at the same time it could be another species. So it's very short. You never can really rely on. So there's a big difference. And then it's very dependent on currents and weather. In the summertime, they're outside when we go deeper down where the water is clean. And it not so much maybe on water quality, but more like different currents come with packages of larvae from somewhere outside in the North Sea that comes in and deposit, and then they sit on there. They need of course the food, and also the food can change during different years. And if there was you have lots of certain food one year, you got lots of nudie. If the nudibranchs are coming in, then suddenly you have lots of nudies. So there's a lot of flux and change in more like here if compared to Indonesia. But then I think we need to study much more what actually is happening in Indonesia, or which species you have and what is actually happening if they have these fluctuations, or and see compare that with water currents or how the water quality. There's so much more to be known about it.
Yeah. Okay. Nice idea to do some research. Okay. Yeah. Maybe Resi, this is last question for you. It's coming from me again. For Nudibranch identification, is there any specific characteristic to distinguish between species? I mean, like in the sponges, we need to check speckles, and in the Stony Coral, we need to check the corallid structure. So maybe for the Nudibranch identification, you have what kind of the specific characteristic?
Yes, thank you, Dr. Anin. For Nudibranch, we need to check the radula. The radula, because the only thing that Nudibranch has is the teeth, the radula. So we bleach them and then we can look that in the SEM for the radula. But first of all, Nudibranch is very easy to recognize from morphology. So it will be okay for morphology. But next we can check from the radula of nudibranch. Yeah, I think that maybe Kenneth, you have another key identification from museum.
Well, you see the general body structure. You see the tentacles, which you call rhinophores. The shape of the serata, these things on the back, or the shape if there's a gill. Some of the dorids have this circlet of gills around the anus on the back, and the shape of that one. And if you have some species that are very hard to separate, because you're going to check the radula, that's this electron microscopy. Take some time. You can also try to look at the have a stylet, the shape of that stylet. So you do it under the microscope. So that could be a little bit quicker than doing the SEM. But then to be extra sure, you can also just check the radular ultrastructure.
Yeah.
Yeah.
If you happen to have an electron microscopy, maybe not everyone has one at home.
Yes, exactly.
You ask Jesse.
Yeah. So it's easy to recognize from morphological first, like Kenneth explained, serata, rhinophore, mantle. And then after that, the last, if we still cannot recognize, we do SEM for radula.
Okay. Thank you very much, Jesse.
Yes.
Yeah. So I think we can move to the next station. That is evaluation and closing. Maybe before we come to it, Kennet, could you please explain to us the responsibilities of curators in the museum? Do they also work with the taxonomy?
To be able to work with the collections, I'm responsible for marine invertebrates. So we had old collections, and we're getting new collections just to keep a collection alive and to make it available. You need to get in more material, new material, and we register in a database. So you can go into the database and see what's there, and making it available on data portals, international, so people can go in and see, okay, what you have here. So I get requests from people all around the world. Yesterday it was some researcher from Scotland, Aberdeen, who is looking for this kind of nudibranch. Do you have anything? And I said, okay, I think 35 specimens from of this millennium. So she wants some samples of them, and also helping European project for eDNA. They're making management of the sea floor, see what species do you have, and do you see any differences? You can do some eDNA. You take a sample and you analyze that, and you find all through the DNA what kind of species you have, but you need to first make a library of different species, and then this mainly German project, they turn to me, we are missing, there's 100 species that we don't have. Do you have them? And then I said, I checked, okay, we have 70 species of these actually from this survey that was made 10 years ago. So these rare species, I said, wow, you can help us. So I was collecting these specimens, and I said, sending them small tissue samples to Germany, where they can do sequencing and use it in the library. So also these less common species get covered if you take a sample of the bottom and then make a screening of it. So then you can have a more effective way of, you don't need an army of specialists. You just, when you have this reference, you can do it automatically. And then when you fall out, here's some strange DNA, it's a new species, they find some new invasive species. So, and then you need to know what you have, and then we are engaged in research research collaborations. So that's the way you learn yourself something where you can find things in the collections, and even if you can't be a specialist on everything, but you need to know with people where the specialists are, and you can help them, and they can help you in the network.
Okay, very helpful. Yeah.
Yeah, and then I also work with the exhibitions also.
Yeah.
So we make text and exhibitions on invertebrates, or now on echinoderms. So we get involved in that and all the public activities also.
Okay.
And then we can, one thing more we can do is we do analysis what was here on the Swedish West Coast 100 years ago, and what do you find today? And then we can compare and see the big changes, what's caused because of Swedish people doing things, humans, and what is natural. That's also one of the big things. So we help the authorities with that.
Okay. So we call it with a Natural History Museum.
Yes, yes.
Yes.
And then we help the authorities to find certain areas with high diversity. That's where you should protect. So now we have several protected areas along the West Coast the last 10 years. That is new.
Yeah, that's important also for Indonesian people.
Yes, you should have marine protected areas. You have some, you should have more.
Okay. Thank you, Kennet, for your explanation. Maybe next series you can show us more pictures about the collection of your museum, so we can file trip, like virtual file trip. Yeah. Okay. Next question for Resi. Could you please explain the Mero Foundation? I mean, the Mero activities. So maybe the students here who come in this lecture can go to Lamben someday for internship in your organization, something. Resi.
Yes, thank you, Anin. Yes, Mero Foundation. Mero Foundation is marine education and research organization. So like Marine Institute. Actually, Kennet also part of the supporting of Mero. So Kennet was there in 2020 or 20 or
Yes.
or 2020.
2019. Time flies.
Huh?
Yeah,
No,
time flies. It was 2020. I was there 2020. Yes.
2020, yeah.
Yes.
Yeah, when we start Mero, yeah. Actually, we start with the Nudibranch, because that's the spot for Nudibranch. But now we develop for many kind of marine invertebrate like sponges, tunicates, hydroid, also platyhelminthes. So Mero provide laboratory for research like scanning electron microscope. So we have SAM Kennet in Mero, and then we have some laboratory like microbiology and also molecular, and we gonna set up for sequencer also. So this is provide for Indonesia, for the student in Indonesia. So we are very welcome for all of you. The student, you can come to Mero in Bali, also diving. So we are working interest for research, many kind of research about ecology, habitat, marine invertebrate, also the diversity, also chemical, also molecular, also bacteria and fungi. And then we are open also for internship and also for research, lecturer or student. And then not only that, we also have like social activity like education to the young generation, teaching English, teaching the kids close to Tulamben area. So we introduce and explain to them about what you can find in the sea, because they only know about fish, and now they know that in the sea you can find sea star and also turtle and also another things. They are living very close with the sea, but they don't know what is nudibranch, what is the sea star. So this is very important for young generation. So that's all of our activity. Also, we are active to apply for grant and also collaboration with the international collaborators. Also with Kennet, we still try to develop and waiting Kennet to come again to Indonesia after pandemic situation. Yeah, I think that's all, Anin.
Yeah.
Yeah.
Okay. Thank you. Thank you very much. Yeah. And then maybe before we end this test lecture, I want to try to summarize your talk, yeah, from the first speaker and the second speaker. That in the first session, I highlight 5 points. There are several films that Kennet mentioned. For the films of sponges, there are 3 classes, such as calcare, demosponges, and then hexatinylate. And for the films ctenophore, there are some genus such as Bulinopsis, Hero, Pleurobrasia. And for the films Cnidaria, there are 6 classes, such as Anthozoa. So if you guys study about corallology, your specimens or your animals include in this class, yeah, hard coral and also soft coral. After that, another class is Cubozoa, Hydrozoa, Scyphozoa, Saurozoa, and Mixozoa. And the phylum Cenozoelomorpha, there are 2 subphylums, Cenoturbellida and Acoelomorpha. And the last, for phylum Platyhelminthes, there is one species. I mean, for the instance, we Kennet saw Prostecereus fetatus. Okay. And yeah, in the second series, second session, I mean, for the doctoracy, the first one, there are a lot of nudibranch that still in the level genus. So therefore, if you guys interest with nudibranch, you also can contribute to study about it. So we can make some identification for nudibranch. And the second, the food of nudibranch is varies from plants like algae to animals such as sponges, bryozoas, soft corals, and so on. For the reproduction, they laying eggs. For the defense, they have glands with acid, camouflage, mimicry, and stinging cells. And for the identification of nudibranch, we need to check radula, gonads, serrata, and also the mantle. Okay, that's all from us. Maybe we would like very thank you for both of you, Dr. Kennet and Dr. Resti. It's nice for us to have all of you. So please stay healthy, and also you can join for the next series. Dr. Kennet will also have another material. So don't forget and don't miss it. We can meet again.
Yes, maybe annelids, crustacea, and echinodermata, those 3 groups.
Okay, yeah. So maybe I can give it back to the Master of Ceremony. Thank you very much.
Okay, thank you, Dr. Anin, for the materials, the lecture for today, for the session, I mean. Okay, so that was the lecture for today's session. So, okay, what do you think, guys? For me, it's very informative and it is important knowledge. Yeah, we gain more knowledge about invertebrates, about like kind of hydrozoa, and yeah, anything else that already explained it by the speakers. Okay, so yeah, for today, finally we get into the last session in this event, which is the closing sessions. And let's close this event by saying Hamdalah. Alhamdulillah. As the host, I would like to say thank you to Associate Professor Kennet Lundin and Dr. Resi as our speaker for today's session, and to Dr. Anin Ditya as the moderator for today. Also for the committee, Ministry of Education, Culture, Research and Technology, Republik Indonesia, also LPDP, which is Indonesia Endowment Fund for Education, for funding. Okay, so I would like to remind you all, don't forget to attend the second series of this guest lecture on 6 October 2021. Also, I would like to apologize if in guiding this event, I made mistakes or offend someone here. Please forgive me. And thank you very much for the attention. I'm Amanda as your master of ceremony, or maybe as a host for today's session, standing by. Bye-bye. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Manda.
Thank you, Mr. Kennet.
Thank you. Thank you, Kennet.
Selamat
Bye-bye.
Thank you,
sore,
Resi.
Mr.
Afternoon.
Thank you, Ani.
Night.
Amanda, bagus.